Okay, for some of you that might already be mad at me, I know meiosis isn't the full process that creates a baby. But I couldn't really think of a more original name, so let's just pretend I never said that and get started. If you don't know what meiosis is, you're watching the right video. If not, you may just be a kid who's doing the same project. Or you might be my teacher that is watching very closely and counting how many bad jokes I make throughout this entire video. So without further ado, let's learn about the process of meiosis. Meiosis is the process that splits a parent cell into 4 new cells that each contain half of the parent's number of chromosomes. These cells are known as gametes, or sex cells, that are used to create an offspring or baby. When those 2 gametes combine to make an offspring, the offspring cell is known as a zygote. However, do not mix up meiosis with mitosis because they are 2 completely different cell divisions. I'll explain these differences later in the video, but all you need to know now is that the major difference between the 2 are the results from their processes. Meiosis is split into 2 phases. Meiosis 1 and meiosis 2. Similar to mitosis, meiosis undergoes interphase for its first stage. Just like mitosis, meiosis replicates its chromosomes during this phase to initiate the entire process. Keep in mind that the organism's parent's chromosomes are being replicated. These 2 colors are very important to remember during the following stages. During prophase 1, centrioles and spindle form, which are the pieces that make the division happen. Then our original and replicated mommy and daddy chromosomes tightly coil up, and then the nuclear envelope disappears. So you might be thinking that this, so far, is a lot like mitosis, but this is where things get crazy. Each parent chromosome actually tightens up even more, connecting each other to create what is known as a tetrad. But it doesn't stop there. The tetrads connect so tightly together that they happen to rub off their genetic codes on each other. This is called crossing over. So for those of you who are lost right now, let me give you one of my bad analogies. Okay, so let's say there are 2 people wearing 2 different colored wet shirts. Why? Because they just got done painting a pink and blue house and they're too lazy to clean themselves. The people represent each chromosome in our tetrad, and each different colored shirt represents our parents' genes. Let's say our guy in the blue is proud of their work and wants to give his bro a bro hug. So they hug it out like bros. And before they know it, they smear their paint on each other. Simple enough, right? Now let's apply crossing over to all this. Remember that each person represented the chromosomes in the tetrad, and that each different colored shirt represented each of the parents' genes. So by smearing their different colors on each other, they're basically transferring their own genes to the other. So the mother's genes are transferring to the father genes, and the father's genes are transferring to the mother genes. This is what gives people their unique traits, rather than being a complete copy of their parents. Now let's move on to the next phase before I fully lose some of you. During metaphase 1, the centrioles build up and send out their spindle fibers into each of the tetrads, centromeres. Then the centrioles move into position at each of the opposite poles of the cell. As they move into position, they move the tetrads towards the equator of the cell. However, unlike mitosis, the chromosomes are situated together along the equator as tetrads, rather than being situated on their own just like they are in mitosis. Why is this important to remember? It's what results from these 2 divisions that separates the 2. Basically, when a tetrad separates in anaphase, it splits into 2 new pairs of chromosomes that are still held by its centromere. On the other hand, when a chromosome of mitosis separates in anaphase, it splits into 2 pairs of sister chromatids instead, destroying the centromere. Normally in mitosis, we cut the chromosomes in half like that, to place each pair of sister chromatids in each new cell. Then within those new cells, the sister chromatids would just replicate themselves, so they could still maintain that same number of chromosomes as the parent cell. But that's not what we're trying to achieve in meiosis, which is why this is important to notice the differences in their chromosomal divisions. Now on to anaphase 1, where the cell actually begins to divide into two. As already stated, the spindle fibers pull and separate the tetrads into two pairs of chromosomes. These chromosomes are pulled into their new cells. There really isn't much more to say about this phase, other than the fact that each cell, as of now, contains only mother chromosomes and only father chromosomes. I mean, I could create another bad analogy for what is happening during this phase. Like the separation of tetrads could resemble medieval dismemberment, right? Yeah? Anybody? No? Too soon? Yeah, let's just go to the next phase. The final part of phase 1 is telophase 1, where the cell divides into two. During this phase, the centrioles and spindle fibers begin to break down. Then the chromosomes begin to uncoil. And the cell splits in half, creating two new cells through cytokinesis, where the cytoplasm splits and pinches itself. With this first division, we now have half the genetic information as we did at the beginning. There was genetic information from the mother and father in the first cell, but now we only have genetics for one parent in each new cell. Now you might be asking yourself, why don't we just stop here if we have half of what we already started with, dummy? Isn't this what meiosis is about? If we end here, both of our gametes, when added together, will still produce an offspring with double the chromosomes. So we need to cut down the chromosomes even more to stop an overdose of chromosomes in the offspring. The chromosome count won't even just add the same amount each time. The amount of chromosomes will continue to multiply over and over again per division. So to even cut down the chromosomes even more, we must move on to the next phase of meiosis, meiosis 2. Before the cells begin to become active again, they go through a short interphase-like sequence. However, unlike mitosis and meiosis 1, the chromosomes do not replicate at all. As prophase 2 begins, the chromosomes coil back up. Then the new pairs of centrioles reappear into the new cells. The spindle fibers from each centriole then connects to each of the chromosomes. You will begin to notice that the following phases are very similar to mitosis itself, so this will be way faster than it was in meiosis 1. So the centrioles move to their opposite poles, situating the chromosomes towards the center independently from one another. So if you remember from earlier in this video, if the chromosomes are aligned alone before they're separated, those same chromosomes will split into two pairs of sister chromatids. So this is what exactly happens in anaphase 2. The sister chromatids separate and are sent to their new cells. Then finally, in our final phase, telophase 2, the centrioles disappear, the cell splits into 4 new ones through cytokinesis, and the nuclear envelope reappears in each of the new cells. So at the end of meiosis, we end up with 4 new haploid cells from our main diploid cell. So before we wrap up, let me explain what a haploid and diploid cell is for those of you who may not know. A haploid cell is simply a cell that contains only one set of chromosomes per parent, which can be represented as N. A diploid cell, on the other hand, has 2 sets of chromosomes per parent, which are known as homologous chromosomes. This can be represented as 2N. Homologous chromosomes may sound like a mouthful, but they are simply just doubled chromosomes that share the same genes or allele letters with each other. But keep in mind that though they may share the same genes, the alleles can differ from each other. Now with all this new information, we can see that our new haploid cells can now live off as gametes and can interact with each other to create an offspring. And since haploid cells are basically a half-down version of a diploid, each human gamete will contain 23 chromosomes. And when they combine through reproduction, female egg gametes and male sperm gametes can create an offspring or baby that will have 46 chromosomes, just like every other human. My name is Adam Vega, and this was the process of meiosis. Thanks for watching.